yes, wherever you go, there you are. But when you get there, you're going to have some friends with you. On the order of 100 trillion friends. Now, naturally, when we think about human beings, it's logical that we would think about us and the cells that make us up. No surprise there. They're actually on the order of 10 to the 13th human cells. But in addition to those 10 to the 13th human cells, as you sit in your seats listening to this lecture tonight, along with you listening are 10 times more bacteria than there are human cells. How can this be? Well, bacteria are small, for one thing. They pack rather tidily, but they're everywhere. If you look at an average small piece of skin, you're likely to find on the order of 1 million bacteria in a square centimeter of skin. If you go into the gut, into the colon, for instance, in a cubic centimeter of the intestinal contents, you'll find up to 5 trillion bacteria. So these are huge, huge numbers. There's another, I think, even more staggering component here, which it isn't just that there are a lot of cells there, but if you actually count up the human genes in our body and compare that to the number of different bacterial genes that are present, we're not outnumbered by a factor of 10, but by a factor of 100. There are 100 times more bacterial genes listening to this lecture than there are human genes in each and every seat in the auditorium. Now, how do we think about that kind of diversity? Well, if you remember the tree of life, you'll know that not all organisms are the same. In fact, if we think about the human genome, the human, and the organisms there are many, many varieties there. In the mouth, there are thought to be as many as 400 different species of bacteria present. In the gut, on the order of 500 or 600 different species. So how do we think about these species? How do we think about biological diversity? Well, again, the tree of life. We live in a part of the tree of life referred to as the eukaryotes. To orient you, there you are. If you're used to going to a zoo and being astonished by all the biological diversity among the zebras and the turtles and the reptiles and the plants, you're looking at about this much of the eukaryotic tree. There are other two major domains in the tree of life, however. The archaea and the bacteria. So I didn't do this necessarily to make us feel puny, but in terms of the diversity, these other two domains, the archaea and the bacteria, are made up entirely of unicellular, non-nucleated organisms. And there are a lot of them, and they do an astonishing number of things, and we know about very few of them. But I want to talk about the ones that live on and in us. Megan already defined the microbiome for us, so I can fly right past that. And instead, talk about the fact that these microbes that are sitting in the seat with you tonight are not simply passive passengers like they might have taken the T to come here tonight or riding a cross-town bus. They're actually intimately linked and influence our health and nutrition. Now, how can that be? Well, here's some examples. Vitamin K, an essential vitamin to human health involved in clotting, we don't make it. We get all of our vitamin K either from the food we eat or from the microbes in the gut that synthesize it. Similarly, vitamin from the B complex, biotin, we don't synthesize it. We rely on the microbes in our gut. Digesting complex polysaccharides. Not many of us, eat corn stalks. Although I was talking to somebody today about this slide, and they said, broccoli, corn stalks, what's the difference? But the point is, human beings cannot digest most of the plant material we eat. Now, we think of termites that rely on microbes in their gut to digest the wood that they chew. And similarly, we rely on microbes in our gut to process the energy in about 30% of the nutrients we get out of our food. There was a particularly enlightening, or interesting anyway, story that emerged on this vein this summer. If you look in the microbes that are present in the intestines of Japanese people, where seaweed has been eaten as a major part of the diet for many, many hundreds of years, they actually found evidence for a gene that is involved in digesting some of the compounds found only in seaweed. And if you ask where did this gene come from, you can map that gene by comparing it to all the other genes like it and find that it actually is normally found in the ocean. And so the idea was that long ago, presumably, people eating seaweed got some of these bugs that also ate seaweed, and in our intestines, the DNA got all mixed up, and that gene jumped into a microbe that was living in the intestines, and it turned out to be very useful there because it helped people who eat seaweed digest the seaweed. So microbes have another really important function, which is as microbial communities, they act to keep potentially infectious organisms at bay. A very common example of this is the pathogen Clostridium difficile. C. difficile is quite a problem in hospitals and nursing homes where people often get prolonged courses of antibiotics. And the impact of those long courses of antibiotics is to deplete and change the microbial communities in people's intestines. And C. difficile will come in and start to grow and cause a really hellish diarrheal infection. And really reflecting the absence of the normal occupants of the gut. You can get rid of C. difficile with antibiotics, but it tends to recur. One of the current therapies for severe C. diff exposures actually falls in the category of something that would not have been considered dinner table conversation when I grew up, consisting of fecal transplants. I mean it. You actually take the microbes out of the feces of healthy individuals and use that to recolonize the intestines of patients with C. diff infections. And it is effective. So again, suggesting that it's the presence of a community of microbes that are serving a function here. Another important role of the microbial communities in and on us, I'll touch on at the end of this talk, has to do with defining our immune system, actually shaping it as we develop, and maintaining the immune system in appropriate balance of readiness versus attack mode. So, I think the point I'm trying to make is that if we want to truly understand human biology, we can't just think about humans, we need to think about the microbes as well. I, along with many of my colleagues at the Broad Institute, participated in the Human Genome Project, the attempt to sequence all of the genes in the human. And it set a milestone in biology of giving us, for the first time, the comprehensive list of all the genes that we work with. The next phase in this exploration of human biology was to define variation. And through the International HapMap project and now the 1,000 Genome Projects, we're really mapping human variation and trying to understand how much do the genes that differ between people play a role in predisposition to disease, response to environmental factors that dictate human health. But we're now in a whole new project, which is, I think, incredibly exciting from the standpoint of understanding human health and from understanding life on this planet, which is the Human Microbiome Project, which is going on right now. Many of the people in the audience are actually involved in this. And this represents essentially a comprehensive census of all of the organisms living in and on us. Now, we've specifically, just as a census, looking at what's found in healthy adults. So how do you do a census? Well, many of us know this quite well because we just participated in the US Census. In the case of the Microbiome Project, we had to pick sites, specific sites to go ask what microbes live there. And so we focused on 5 major body sites. And from a large 250 individuals who are going into the clinic to be sampled, we're taking what the contents of either by swabbing or spitting or various bodily functions that deposit samples in containers. We're asking, what are the microbes present there? Now, this is actually kind of an interesting thing because how do you do this in a controlled fashion? Well, they had to spend many, many months, as you can imagine, planning. How do you actually make sure that everybody coming to have their microbiome sampled didn't, for instance, take antibiotics last week or use some powerful new antibacterial soap or gargle with Clorox the morning that they came in? So the protocol for setting up this has been very highly regimented. And for the first time ever, we're collecting information not just about the bugs or the people, but all about the people's health, their health status. And we're out not only just sampling them as they come in today, but many of these people will be sampled over time. So we'll get repeat visits and we can ask, how does the microbiome change over time? So it's astonishing, but this is the first time ever we will be able to ask questions as fundamental as how different are the microbiomes of different individuals? And how stable is that? And what does it possibly mean? So now back to the idea of doing this census. Well, US Census Bureau mails out things to every household and they wait for them to be mailed back. And they found that this is a little flawed because, of course, not everybody mails their census form back. In fact, some people don't have addresses to even mail the form to. Well, the microbial equivalent of this is that for years we've done microbiology by going to locations, either on us or in the soil or in the environment, and trying to culture those microbes on Petri dishes. So here we see a scientist holding up a Petri dish, and it's covered with bacteria. And we can describe the bacteria on those Petri dishes. But unfortunately, when you go into, for instance, the human gut, 80% of the organisms in our gut will not be cultured, have not been cultured. So we have no method to study them. So what we've had to do is come up with a new method to take the census of bacteria that does not rely on culture. So fortunately, DNA sequencing technology has made great leaps, spurred by places like the Broad Institute, where we have a sequencing center that can use high-throughput sequencing technology to do these census. Now, how do you actually do this efficiently? How do you find microbes? Well, you need something you can go look for. And in the 1970s, Norm Pace and his colleagues realized that one of the most useful things to recognize bacteria is the ribosome. Ribosome is a complex structure that makes proteins, and all cells have them. And they don't change very rapidly. So all ribosomes in bacteria look roughly the same. I say it's essential and it's complex. It's complex because it's made up of RNA, which is the sort of orangish color here, and many, many different proteins. On the order of, what, 21-something proteins in the small subunit of the ribosome. So who cares? Well, it's a complex thing, and evolution finds it very difficult to change such a complex mixture of proteins and RNA. If you change one thing, the whole thing isn't going to work. I'm reminded of when we were rushing off to go out to dinner, and my daughter spilled something on her skirt. And I said, well, go change your skirt. And she gave me this look, Dad, I can't just change my skirt. If I change my skirt, I have to change my top. If I change my top, I've got to change the shoes and the handbag, and this belt's not going to work. And then I've got this headband thing. Dad, it's an outfit. The ribosome is an outfit. And it can't change quickly, even over evolutionary time. Making proteins is so essential, it always has to work, and it's very hard to innovate. So What you see here is an alignment of DNA sequences that all come out of different bacteria. Now, you might think, all right, they're all different bacteria, they must be similar, but these come across these huge reaches of evolutionary time. And many of the sequences between these different ribosomal, many of the bases in the different ribosomal sequences are identical between all of these bacteria. Who cares? Well, it means we can use those common pieces as essentially a hook to go in and grab bacteria that we know nothing about because we know they have those sequences. We can pull them out using that hook, we can sequence them, and then we can ask who was there. We never had to culture them, and we can do our census. And that is exactly what we've been doing. So, what have we learned so far? Well, first thing we've learned is that if we go in and we start pulling out bacterial sequences, we ask, do we recognize this organism? And this is just like census taking. Are they, what's their last name, basically? Who lives at this residence? Are they Smiths? Fine, oh, they're Smiths. How many Smiths live in this house? And that's what we've done here. So each column represents a site of the human body. Nose, gut, skin, for instance. And anywhere a particular bacteria is found, we color it in. And the more we found there, the brighter the color. So if there's red, there's a lot of it there. And the first thing you can see is that pretty clearly, body sites look different from each other. You can zoom in and get an idea of this. For instance, the mouth has certain kinds of bacteria that really don't ever show up in the gut. Mouth, gut, but what's in our mouth we swallow, it ends up in the gut. How does that work? It works. It's different. Distinguishable microbiomes in different body sites. Fancy word for nose. Nose looks a little bit like mouth, that makes sense. It looks a little bit like skin, that makes sense. So distinct body sites have distinct microbiomes. If you step back and look at this now, you can see, for instance, there are certain kinds of bacteria we never see outside of the mouth, the oral cavity. They're very, very rare elsewhere. And so there is a distinct signature of the microbiomes of these sites. So we would say that body sites have distinct microbiomes, but there is person-to-person variation. Where is there person-to-person variation? Well, right here. Each column is a different person. Each little square represents a different person. So there is some variation. Well, if we take all of that information about who was present and how many there are and collapse it down to a signature, we can come up with a computational way to compare different communities of bacteria and compare these different samples. And so what am I showing here? I'm showing a comparison of the microbiomes found in different people by different sites. So in green are all of the microbiomes that came out of a nose, somebody's nose. And you can see they largely cluster together. All the red ones represent samples that came out of a mouth, and you can see there's very little confusing the samples that came out of the mouth and what came out of the nose. And yet, there's a lot of variation between what's in the mouth. Let's spin that again. There's the stool hiding over here. Can I back it up? No? Anyway, so there's already, this is just data based on the first 18 people that came through the clinics. And already we're starting to see real distinct differences. Pretty remarkable, I think. There we go. We do get to see it again. There's the stool over there. So this idea of taking a census clearly works. Well, what do we do with this information? We can ask more sophisticated questions. Like, what about local environments? Do they differ? So here I've described something that's essentially 2 millimeters away from each other, tiny, tiny distance, the gums and what's under the gums. And again, you can see clear separation of the microbes found in those two different environments. Even more startling is the fact that you can actually go into different teeth side by side and get reproducible, distinct communities. So when we think about ecology and, gee, the rainforest and all of the exciting exotic places that things can live, we have to start thinking about the distinct ecological niches in the human. Now, you're probably all desperate to run out and brush your teeth at this point. But that was describing the differences in normal, healthy adults. The question we have is, can we recognize differences in microbial communities in disease states? And one of the first states we've looked at, we, in the consortium doing this microbiome work, is in the colon, and the diseases being ulcerative colitis and Crohn's disease, inflammatory diseases of the large intestine. And if you simply take stool from normal adults or patients with either of these two diseases and ask, do you find different microbes present in them? The answer is absolutely yes. No pretty colors here, but it's the same analysis that says healthy adults have a certain composition, typically, of their microbes in the stool. If you go to ulcerative colitis patients, you find overlap. There's not a complete separation, so there's some similarity, but different enough. And if you go to patients with active Crohn's disease, the microbes you find in their stool are fundamentally different. That's pretty powerful. Now, unfortunately, I could not begin to tell you what's cause and what's effect here. Right? And this is something that scientists labor a great deal over. We never want to confuse causal relationships with simply correlations. As my wife will tell you, I tend to sing every morning and the sun comes up. I'm not confused that my singing actually makes that sun come up. So, in this case, this is simply, gosh, who would have thought? Are there cases when we can go in and ask, do we see a causal relationship? And the first place people saw this was comparing the microbes in the stool of lean and obese humans. And what I'm showing here is two large groupings of bacteria. Normally, these two make up most of the kinds of bacteria in our gut. And what you can see is a shift in the proportion of these two different kinds of bacteria in either obese or lean individuals. Now, it's not a complete replacement. It isn't that some things just disappear and others appear, but this satisfies any statistician that this is a real difference. If you take these obese adults and put them on a diet, in this case for 1 year, and it didn't matter what kind of diet, and they lost weight, what you saw was a gradual shift from the signature of the obese to the signature of the lean individuals. That's pretty cool. Now, again, this is not cause and effect. Hard to do those experiments in humans. Not sure we would find it ethical. But we can do studies like this in animals where we think the implications will hold true as well. And most typically, we do this in mice. What's interesting about mice is you can actually raise mice in a germ-free environment. How do you do that? Very carefully. No, C-section. You deliver them by C-section, and you raise them with sterilized food, sterilized cages, sterilized everything. And they grow. However, if you take the normal contents of a mouse intestine and introduce it into these germ-free mice, suddenly they eat less, but they gain more weight. So the microbes in the intestine are helping them harvest energy from their food more efficiently. There's a genetic model of obesity in the mouse. If you take the microbes out of the intestines of the obese mice and you give them to their lean litter mates, what you find is that those lean litter mates then start gaining weight much faster than they otherwise would. So that's causal. You can say the microbes in the obese mice actually confer the ability to gain weight faster, which is pretty darned amazing to me. So how can that be? How can microbes do this? Well, we would say, being a geneticist, it's in the genes. It's not culture, it's in the genes. And if you ask about the kinds of genes that are different in these two kinds of organisms, you actually do find that the firmicutes, which are the ones that are found at higher abundance in the obese humans and in mice, contain more genes that allow these organisms to capture energy, break down sugars, and presumably lead to weight gain. Now, it's not quite that simple, because there are actually two things going on. One is what I described, is that the microbes make it more efficient for us to harvest energy from the food we eat. But there's a whole nother pathway. So that's this. Microbes just eat our food better and produce it, allow us to digest it better. But there's some signaling going on where the microbes actually send signals to the normal regulation of body weight in the mouse. And these microbes actually get the mice to turn on fat storage. So pretty darn complicated and pretty darn cool. These microbes really do influence the physiology of these animals. So if we are going to understand our microbiome, we better understand the genes found in these organisms. And that's exactly what the Human Microbiome Project is also doing. I described the fact that we're sampling normal healthy adults and finding out what's there and doing the census. We're also sequencing genomes from organisms that are normally found on us. Now, you would have thought perhaps that all these genomes would have been sequenced before, but that's simply not true. In fact, we just published a paper now 2 months ago describing 178 new bacterial reference genomes coming out of human. And in doing that, we discovered from the maybe 500,000 genes that were present, 30,000 of these genes were completely novel, had never been described before. So obviously, we have a lot of biology to unravel by sequencing these genomes and figuring out what they do. And it's kind of one of the most exciting parts of my career to think that we're not only understanding the human genome and human variation, but starting to understand the variation within the microbes that live within us. So, okay, we've got microbes. Where do they come from? Said you may all want to go back and brush your teeth ferociously, but I guarantee you'll have microbes there in the morning. Where do we start with? Where do our microbes come from? Well, this is probably pretty obvious. Like everything good, it comes from mom. So a study was just published showing this quite conclusively, that newborns, if you look at their microbiome, how they come into the world determines what you find there. What do I mean? Well, this was a study that took about 8 children born vaginally and 8 children born by C-section. Before the children were born, they did a lot of swabbing of the mom, skin, mouth, vagina. 5 minutes after the baby was born, they did a lot of swabbing of the baby. And they compared what did they see. And what they saw is pretty striking. That is, the mother's oral community is up here. None of the kids look like the mom's mouth. But the children born vaginally look very much in terms of their community composition like the microbes found in the mother's vagina. The kids born by C-section have microbes that look very much like the mother's skin. So this is literally 5 minutes after birth. They went on and looked at other time points. They looked at meconium and found basically this same thing. But it isn't just that everybody gets the same microbes. It really is that you look like your mother. For instance, here is baby 1. And what you find on baby 1 compared to what organisms were found in that mother's vagina. Very similar. Take something very different. Here's baby number 4. Here are the microbes found in baby number 4's mother's vagina. So you get this stamp at a very early age. Now, I should hasten to say that these aren't necessarily the microbes that persist. If we want to know about where our microbe comes from, we have to follow children over time. And during the first year of life, it fluctuates dramatically. But by the time babies are about a year old, they start to have a microbiome that looks rather adult-like. Now, how that gets established is still not entirely clear. But it is obvious that there are many contributing factors, including environmental exposure, what you eat, and your own genetics. So this is brand new stuff. I said we were just trying to profile adults for the first time. This study I just pulled down online. It hasn't come out in paper yet. But it's very exciting because clearly we're very interested in how you establish a normal microbiome in children to understand adult health and development. So I've been talking about the GI tract as if it was plumbing. It isn't simply plumbing. That is, it's not pipes with water flowing through it. They're living cells on both sides. They're living cells of the bacteria on the lining of the gut, and of course the human cells that make up the gut itself play an active role in this. And signals are going back and forth all the time between these two types of cells. So rather than think about the human gut as a pipe, I'd like you to walk out of here thinking tonight of the human gut as the single largest immune organ in the body. That's not how I usually think about my gut, but that's the right way to think about the gut. There are a fantastic number of specialized cells in the human gut involved in immunity. And why is this? Well, it makes sense. What I've shown here are human cells in the darker color on the bottom, and the intestinal epithelium over here, and then this would be the lumen, or the place where all the bacteria and the food particles are. So that's a really, really interesting boundary. You have human cells in direct contact with bacteria. And so the immune system has all kinds of stuff going on at this boundary. Like what? Well, they're cells that make specialized antimicrobial peptides, defensins, for instance, that are actually secreted and control the growth of certain bacteria. There are other immune cells that actually recognize specific bacteria. There's some here that are making antibodies and pushing antibodies out into the intestine. And you can see here an electron micrograph of the exquisite interaction of bacteria and human cells. What's shown here is a bacterial cell, in this case Helicobacter pylori, which is known for as a cause of ulcers and gastric cancer even, in this remarkable association with gastric epithelium. It induces this special structure, this pedestal actually forms, and then molecules move back and forth between these cells. So again, it's not a pipe, it's not things are just flowing through. Things stop, things meet, they adhere, they talk to each other, and they have an incredibly complicated conversation. This is data I borrowed from Romnick Xavier, who's an associate of the Broad at MGH, showing some of the exquisite nature of this conversation, just looking at the expression of certain kinds of proteins in macrophages, immune cells. And you can, what I've shown from Romnick is different bacteria exposed to the same cells turn on very different sets of genes. So again, the immune system isn't saying, ack, bacteria. It's exquisitely picking through the pile and saying, ah, here's a bacterium I recognize, and I have a specific response to this one. Here's a different bacterium, and it will get a distinct response as well. So incredibly sophisticated. We go back to this drawing, and I think the important thing that I take away from this is that the immune system has a couple of different ways of interacting with what it perceives as threats. Some of the right way to react to threats is an acute response in which you sort of ring all the alarms and call out the fire brigade and attack. And in other cases, you simply want to hold things in check for a while, and you don't want to overdo it. And there are these two different branches of the two different cell types that actually control this balance of pulling the fire alarm versus saying, no, let's take a more measured response. So there's regulation of the immune system. It isn't simply that it's always in attack. There are some cells that actually turn things down. And that's all going on in the gut. So complicated stuff, immunity and microbes. But what we're learning from all of this is I think that it was naive to imagine that we can classify all bacteria into piles of good or bad. So again, here's another slide from Romnick talking about the fact that commensal bacteria, those that can live in you and not cause any harm or in fact do some good, as opposed to pathogenic, infectious, or bad bacteria, may not be simply a matter of absolutes, wearing a black hat or a white hat. It's a progression. It depends on where they are. For instance, bacteria that live in your gut quite happily can do terrible things if they get outside of that. Would you call it a pathogen? It depends where you find it. It depends on the timing. When do you find this bacteria? What's going on with your immune system? Is your immune system revved up due to other factors? If so, it might overreact to something that would not ordinarily cause a problem. And of course, your genetic background actually also dictates how you perceive a specific bacterium. So there are both microbial and host factors that determine whether a specific bacterium can be thought of as the good guy or the bad guy or a neutral guy. Similarly, immunity, our immune system, isn't simply thinking of a sleep or attack. There's actually part of the status of being on alert in which it's conferring protection to you. However, certain disorders, the immune system gets too active, causes immune pathogenesis, that is, a disorder of the immune system that leads to disease. So the conclusion here is that a pathogen, a specific bacteria or a microbe, may be a pathogen or it may be a commensal, depending on factors beyond its control or beyond the bacterium. So this leads me to sort of a well-noted change in public health over the last 60 years. That is, the astonishing rise of a number of inflammatory and autoimmune disorders. We're familiar with, this is shown 1990 to 2001. That's just 11 years, the prevalence of asthma, that's in the United States. Similar graphs for diabetes going back to 1960. And then to here, 2010, with the projection into the future. Eczema and hay fever. These trends are noted not just in industrialized developed nations, but in the developing world as well. So people have been scratching their heads about this for a long time. What is it that could explain dramatic change in something as fundamental as the incidence of a disease like asthma? The one thing we know is it's not human genetics. Our genes have not changed that rapidly. This has happened in a matter of 1 or 2 generations, so it isn't us. It must be something in the environment. Now, a lot of things have changed in the environment during this time. And there are many, many theories. You grab your favorite one about the environmental changes. In Boston, we like to think about all those years when the Yankees were winning. How could anyone stay healthy under duress like that? But is it environmental? It must be. What in the environment? Well, one of the things we know that differs between then and now is our fixation with fighting microbes. The use of antibiotics became prevalent after World War II in this same time frame. The control of infectious disease, probably the most important thing that humans have ever done to change the course of human health. But that's happened in this same time frame. So is there a possibility there's a link between infectious microbes and some of these inflammatory and autoimmune disorders? Well, people have proposed this. It's referred to as the Hygiene hypothesis. It was first noted in 1989 that if you just look at the incidence of hay fever, this was in 17,000 children born in 1958 in England. The incidence of hay fever went up, well, so turn it the other way around, went down. There was apparently a protective effect from having lots of siblings and having lots of older siblings. And that was also ultimately seen for multiple sclerosis, type 1 diabetes. Another observation, same thing with hay fever and other atopic disease, exposure to farming early in life, exposure to animals, cow sheds, all of that stuff. If the mother was exposed when she was pregnant, the incidence of these allergic diseases went down. So daycare, kids who go to daycare in their first 6 months of life, again, asthma, atopic dermatitis, much less frequent. So, again, not cause and effect. But the hygiene hypothesis is asking, do some infectious agents help protect against immune-related disorders? So here we have to stop and think about what the human immune system really evolved to do. It evolved to keep us alive in a really nasty, hostile world full of bugs, dirt, and lack of hygiene. And it was tuned over those many, many hundreds of thousands or millions even of years to help us stay healthy when exposed to organisms that wanted to do us harm. For instance, if you trace back in evolutionary time when our adaptive immune system is, that is, when the part of the immune system that recognizes insults and adjusts in our lifetime to them, we were already being infected with things like worms at that point. So our immune system evolved in the presence of these organisms. So certainly, there are lots and lots of lifestyle events and environmental factors that have been going on during this same time period. If you look at a graph of the incidence of some of these diseases in Europe, for instance, you can draw all kinds of correlations. The one I liked the most was the one that shows that dinner time, actually, what time families eat dinner, actually correlates with the incidence of some of these diseases. On a north-south basis, of course. There's some animal models that are very, very suggestive, however. For instance, the non-obese diabetic mice. the frequency of diabetes in these mice depends on cleanliness, but not in the way you would initially expect. If you raise these mice in germ-free environments, they almost all get diabetes. If you raise them in dirty housing, they do not get diabetes. Furthermore, if you take the germ-free mice and add bacteria, you can protect them from getting diabetes. This is true in many, many other animal models of autoimmune and inflammatory disease as well. So we've got some big questions in the microbiome still to answer. Among them, do we share a core microbiome? If you compare people, is there something that's common to all of us? How does it vary, for instance, with the way we eat, with our age, with our geography, with the use of antibiotics? Can we read our microbiome out in a way to learn about our health? And of course, the big one, can shifting our microbiome actually lead to changes in our health? To do this, we're going to need a lot of new technology. The Broad is a place devoted to genomic technology. I really do think of this as exploring new worlds. We need to start culturing these unculturable organisms. We need to start sequencing individual cells so we don't have to worry about culturing them. We need to be able to sequence lots and lots of communities at once and all of the genes in them at once. And these experiments generate huge amounts of data, and we need new processes capable of dealing with that. So to summarize, we're part of a complex community of interacting organisms. You cannot think of it strictly as us and them. You can't think of a single bacterium in isolation from the other bacterium that it talks to, and you can't really think about us absent those other organisms. Specialized communities give different functions in the different body habitats. We don't understand this very well yet, but we will. Human health requires a balance of organisms. And there are many, many cases where it's thought that it really isn't the arrival of a single bad bacterium, but it's a community getting out of balance. It's an ecological problem. We can't understand human health without understanding these microbes. Once we do that, what is possible? I think a lot is possible. Clearly, the fact that you can distinguish a patient who has active Crohn's disease from somebody with ulcerative colitis based simply on the microbes on their stool says there is diagnostic power in microbes. Brand new area of science. We know nothing about it yet. Could we build therapies based on this? I believe so. I would like to pause here, stop, and thank, first of all, a lot of people at the Broad Institute who work on the microbiome with me. I'm quite pleased to share this excitement with these colleagues. The Broad Institute is led by Eric Lander, who has pioneered many of these efforts to both understand humans and microbes as models. We're doing this in conjunction with a large consortium. And I just want to point out that many, many different institutes at NIH put money into this project, because every single NIH institute felt that understanding the role of microbes in, for instance, heart disease or diabetes or blood disorders or all of the different institutes, cancer, is one of the most important things we can do. Thanks so much for your attention. I would be delighted to take questions.
It's known in the evolution of bacteria that the more bacteria species that you sequence, the more proteins that cannot be characterized. That means each organism, once you sequence it, about 10 or 15% of the proteins cannot be matched to anything. They're usually called orphans. They're usually smaller and not as big as the regular proteins. Is that what you're finding in your 30,000?
Could you hear that? No. Okay. Let me paraphrase. So traditionally, when we sequence new organisms, we get very excited to look at the DNA sequence, and then we scratch our heads over a great deal of it and say we don't recognize what these things do. And the question is, as we keep sequencing more and more bacteria, do we keep finding more and more novel things? And the answer is absolutely yes. At this point, it's like, coming to a new continent and collecting animals, and they're all things you've never seen before. In time, we expect they will start being familiar, but at this point, each new genome we sequence has on the order of 10 or 15% things that make no sense to us, and yet they clearly do things that matter to us.
I think I can do this the opposite. I realize that you talked about not knowing cost, but has there been any thought given to doing this fecal transplant thing with people with ulcerative colitis, as well as this, so just to see if doing the fecal transplant would cause them to have direct population of microbes?
So the question is, if Crohn's disease or ulcerative colitis, for instance, we know that these patients have a shift in their microbes. Even if we don't know the causal relationship, could we try and treat it by fecal transplants or other things? Well, among the other things that get mentioned most often are probiotics. That is, giving mixtures of organisms that we think will make it through the gut and recolonize, and that's a very active area of research in both UC and Crohn's. Fecal transplants, I believe, are also in the works. I don't know any results from that, but yes.
When you say that you sequenced 30,000 new genes, one thing I've never understood is what makes a specific sequence of genetic material a gene?
Great. Thank you for a very excellent question. What's a gene? And why do I say I found new ones? A gene, typically, we think of a gene as the genetic information that encodes a protein. The protein will be the thing that does most of the work in our cells. So typically, we were raised to say a gene makes a protein. Because it's biology, it gets more complicated than that. But typically, when we say we found 30,000 brand new genes that have never been seen before, we're saying we found the capacity to make 30,000 new proteins that are unlike anything we've ever laid eyes on before.
You mentioned earlier that Japanese people have a sort of gene to digest seaweed that's not seen in other places, and you said that you believe it came from microscopic organisms in the ocean. Would it be possible then to modify people's genetic codes and actually change their DNA by introducing new microorganisms?
Okay, so the question is, if people in Japan have a microbe in their gut that helps them digest seaweed, can we change human genes by introducing microbial genes? So, hot topic of debate. But in the first case, we actually changed human health, not by changing the human DNA, but by changing the DNA of the organisms in us. And let me digress for a moment to introduce this notion of metagenomics, a term I haven't used before. But I first learned of it from ecologists across the street who study things in the ocean, large communities in the ocean that all have to work together because some organism does the work of harvesting the light and turning it into, from the sun, turning it into something that other organisms will eat. Those organisms produce something that something else eats. And as a community, it works. And the term metagenomics means that you really have to think about all of the genes in all of those organisms. I'd like to use this metaphor of a potluck dinner where somebody brings a dish and you get to eat everything else. And it doesn't actually matter which organism has which genes. As a community, as a potluck, it works. So in our guts, to go back to the question about the genes that allow people to eat, digest agar, that's a community that's part of you. You didn't actually need the gene to be in your genome to be effective. One of the things people have thought about is, could we use these microbes to produce drugs? We wouldn't have to introduce the gene into the human genome, but we could simply, take a dose of microbes that make something we need. So I think the simple answer is we don't actually necessarily need to put all of those new genes we want into our own genome. We can put them into the microbes in our guts. Way up top. Yeah?
I said that sound or something strong
Yep.
What happens is like they have a certain response, right?
Yep.
What happens if the response to some other enzymes?
What happens when the immune response gets confused? Yeah. So the question is, if we have such exquisite responses to certain microbes, how come we don't, what happens if you, so one of the articles I was reading used the analogy of warfare, and of course in any warfare there's collateral damage. Sometimes you shoot someone on your side. That is exactly going on in the immune system. And some of the diseases I described, that's exactly it, this inflammatory response, this over-response, the autoimmune response that starts out going after one microbe and ends up wiping out a whole division of the good guys, is exactly that immune recognition gone wrong. So it happens and it's a problem. Go ahead.
Now, you mentioned how that is a great immune system by itself. When we are now ingesting meat, for example, that has been pre-antibiotic, I guess, and so you're carrying that chain along. Do you have any predictions about what could happen to the human immune system as a result of this long chain, not just with killing all the germs with other things, but in our food itself? Does that make sense?
Yes, it makes a lot of sense. So the question is, if the human gut is an important immune system, what happens when we start eating foods, meats, grains, agricultural products that have antibiotics in them? And what's the long-term consequence of this? I will state quite frankly, I don't know the long-term consequence, but it's pretty clear that there's a short-term consequence to the heavy use of antibiotics in agricultural products, namely, anywhere you use an antibiotic, eventually you get organisms that become resistant to it. And you can quite easily start to see how those organisms don't stay in the chickens that they were treated originally in, or the other agricultural products. And those organisms could move into humans. I should say, this Deb Hung, colleague at the Broad in Infectious Disease, just came back from a meeting about antibiotic resistance in Europe, where this is a huge problem. It's not much paid attention to in the United States at the level it is in Europe. Antibiotic resistance and the overuse of antibiotics and the consequences for antibiotic-resistant bugs moving from the food supply into us is a huge deal there. She said they have public billboards talking about not overusing antibiotics. But to go back specifically, what is the likely impact? Honestly, we don't know. With 500 different organisms, we do know, for instance, that people have started to look at how does the gut microbiome respond after antibiotic treatment. And most bugs, microbes, come back very quickly. Within a couple of weeks, most of them are back in roughly the proportion you saw before the antibiotic treatment, but not all of them. Some take many, many months. What's the impact of that? We don't know, but if my metaphor of the potluck dinner is appropriate, it means that sometimes you're going to have a party where no one brought cups. And it isn't simply the number of organisms that matter. We know this from field work in ecology. Sometimes organisms that are there at very, very low numbers actually provide a key pathway in the biochemistry of the whole ecosystem.
So we're going to take one last question.
Thanks. So a lot of the diseases that you talked about today have been studied from a human genetic point of view, and a lot of variants in the human genome have been associated with the prevalence of these diseases. So did you find that the same variants associate with the variation in the human microbiome?
Okay, so the question is, I talked about human diseases, and we know that their variation in the human population leads to differences in the predisposition to some of the human diseases. Can we yet correlate that with any aspect of the microbes that live within us? And the simple answer is not yet. Not yet. I think those studies will be done much sooner in mice, where you can control the genetics and the microbes easily. But when we take these 250 people and we sample them multiple times, in all cases we're taking blood from them, so that if we start to see differences, we can ask about the human genetic basis for the difference in the microbes. For instance, people with a strikingly different microbial composition in the mouth, who may have reduced likelihood of gum disease, those would be exactly the kind of people we want to go genotype. We want to ask what's in it in the human genome that may underlie that.
So before we thank Bruce, I just want to make two quick announcements. One is that I'm sure that Bruce will be willing to answer a few questions if you want to come down with individual questions. The other is that we have a reception out in the lobby, so you're welcome to go to the reception. And just so you know, I just want to repeat that we are skipping next week. There's no lecture next week, but the next 3 Wednesdays at 6 o'clock, we have our other 3 lectures for the series. So let's join me in thanking Bruce.